Chapter 10. Bile flow and fat emulsification. Bile flow determines whether dietary fat can be transformed into a form that enzymes can access and the intestine can absorb. Lipids enter the digestive system as large, hydrophobic aggregates that resist interaction with the aqueous environment of the gut. Without structural modification, these aggregates limit enzyme contact and prevent efficient breakdown. Bile changes this condition by dispersing fats into smaller, organized structures that increase surface area and allow lipolytic enzymes to engage. The effectiveness of this process depends on both the availability of bile and the timing of its release relative to the arrival of fat in the small intestine. Bile is produced continuously but stored and concentrated until it is needed. Its release is triggered by signals that originate as fats move from the stomach into the upper small intestine. These signals coordinate gallbladder contraction with pancreatic enzyme secretion, ensuring that bile and lipases are present at the same time. When this coordination is intact, fats are rapidly dispersed into smaller units, and enzymes can cleave them into components that can be transported across the intestinal lining. The sequence relies on this alignment. Bile must arrive when fats require dispersion, not before and not after. The structural role of bile is central to fat processing. By organizing lipids into smaller particles, bile increases the interface between fat and the surrounding fluid, creating conditions where enzymes can access and cleave triglycerides effectively. Without this dispersion, lipases encounter large, compact fat masses with limited surface exposure. Enzymatic activity becomes restricted to outer layers, leaving internal portions unprocessed. The difference between dispersed and aggregated fat determines how much of the dietary lipid load can be reduced and absorbed. Upstream conditions influence how well bile can perform this role. When gastric processing exposes fats and delivers them into the intestine in a controlled manner, bile can interact with substrates that are already positioned for dispersion. When fats arrive in larger aggregates due to incomplete preparation, or when gastric emptying is irregular, bile delivery may not align with the substrate. Portions of fat may pass through without adequate emulsification, reducing the efficiency of breakdown and absorption. Timing remains the critical variable. Signals that regulate bile release depend on the detection of fat within the intestine, but these signals are influenced by earlier stages of digestion. Weak early-phase signaling or disrupted gastric coordination can delay or reduce the strength of these signals, leading to bile release that is insufficient or mistimed. When bile arrives too early, it may not coincide with the presence of fat. When it arrives too late, fats may have already progressed beyond the optimal region for emulsification. In both cases, the interaction between bile and substrate is reduced. The consequences of impaired bile flow extend beyond fat absorption. Lipids that are not properly processed continue through the intestine, altering the luminal environment and influencing how other nutrients are handled. These unprocessed fats can affect motility and interact with microbial populations, contributing to variability in the digestive sequence. The system attempts to compensate through adjustments in motility and secretion, but these responses do not restore the initial alignment required for efficient fat processing. Bile flow and fat emulsification, therefore, represent a coordinated process that depends on structural preparation, signaling, and timing. Bile does not act in isolation. Its effectiveness is determined by how well the system delivers fats into the intestine and how precisely its release is aligned with that delivery. When these elements are synchronized, fat digestion proceeds with consistency. When they are not, the efficiency of lipid processing declines, and the effects propagate through the remainder of the digestive system. Bile as a structural fluid, not just digestive. Bile functions by changing the physical organization of lipids rather than simply assisting in their breakdown. It acts as a structural agent that reorganizes fat within the intestinal environment, converting large, hydrophobic aggregates into dispersed particles that can interact with enzymes and the surrounding fluid. This transformation determines whether lipids can be processed and absorbed. Without it, fats remain in forms that resist enzymatic access and limit transport across the intestinal lining. The amphipathic nature of bile components allows them to interface between fat and water, creating stable structures that increase surface exposure. As bile interacts with dietary lipids, it breaks larger masses into smaller units and stabilizes them within the aqueous environment of the intestine. This increases the contact area available to lipolytic enzymes, allowing cleavage to occur across a broader surface rather than being confined to the outer layers of a compact fat mass. The structural arrangement created by bile therefore defines the efficiency of enzymatic action. This role extends beyond initial dispersion. Bile maintains the organization of lipid-derived products after enzymatic cleavage, forming transportable structures that carry these components to the intestinal lining. These structures move through the luminal environment in a way that allows consistent interaction with the absorptive surface. Without this organization, the products of fat digestion would remain poorly distributed, reducing the likelihood of efficient uptake. The effectiveness of bile as a structural fluid depends on timing and substrate condition. Lipids must be presented in a state that allows bile to interact with them fully, and bile must be available when this interaction is required. When upstream processing has exposed fats and gastric emptying delivers them in controlled amounts, bile can reorganize them efficiently. When fats arrive in large aggregates or when bile release is misaligned, this structural transformation is incomplete. Portions of the lipid load remain resistant to dispersion, and the efficiency of both enzymatic breakdown and absorption declines. Bile also influences the broader environment of the intestine. By organizing lipids into smaller particles, it affects how contents move, how enzymes distribute, and how other nutrients interact within the same space. This structural influence extends to the consistency of the luminal contents and the pattern of contact between substrates and the intestinal surface. The physical state of the contents is therefore shaped in part by bile activity, linking lipid processing to overall digestive dynamics. Viewing bile as a structural fluid clarifies its role within the digestive sequence. It does not merely support digestion in a general sense. It determines the physical form of lipids and their derivatives, and that form dictates whether enzymes can act and whether absorption can occur. When this structural function is aligned with upstream preparation and downstream demand, lipid processing proceeds efficiently. When it is not, fats remain in forms that limit interaction, and the effects extend through the remainder of the digestive system. Emulsification and micelle formation. Fat digestion depends on converting large lipid aggregates into dispersed, transportable structures that can move through an aqueous environment and interact with the intestinal surface. Emulsification is the first step in this transformation. Bile interacts with dietary fat and breaks it into smaller droplets, increasing the total surface area exposed to digestive enzymes. This is not a passive mixing process. It is a structural reorganization that changes how lipids behave within the intestinal lumen, allowing them to remain suspended rather than separating into larger, inaccessible masses. As emulsification progresses, lipolytic enzymes act on the surface of these droplets, cleaving triglycerides into smaller components. These products do not remain randomly distributed. They are incorporated into organized assemblies known as micelles, which are formed through the interaction of bile components with lipid breakdown products. Micelles act as carriers that keep hydrophobic molecules dispersed within the aqueous environment of the intestine. Without this organization, these molecules would tend to reaggregate, reducing their availability for absorption. The formation of micelles determines whether lipid-derived components can reach the intestinal lining in a form that can be taken up. These structures move through the luminal space and deliver their contents to the surface of the epithelium, where absorption occurs. The efficiency of this delivery depends on the stability and concentration of micelles. When emulsification is complete and bioavailability is sufficient, micelle formation proceeds consistently, and lipid transport to the absorptive surface is maintained. Upstream conditions influence how effectively this sequence operates. Lipids must be presented in a state that allows bile to interact with them, and the timing of bile release must align with the arrival of fat in the intestine. When gastric processing exposes fats and delivers them in controlled amounts, emulsification occurs efficiently and micelle formation follows. When fats arrive in larger aggregates or when bile release is delayed or insufficient, emulsification is incomplete. This limits the formation of micelles and reduces the delivery of lipid components to the intestinal lining. The interaction between emulsification and micelle formation also affects the overall environment within the intestine. Properly dispersed lipids allow enzymes to act uniformly and maintain a consistent pattern of breakdown. When dispersion is incomplete, regions of the lumen contain lipids in different states, creating variability in enzyme activity and transport. This variability influences how effectively nutrients are absorbed and how stable the digestive process remains. Emulsification and micelle formation, therefore, represent a continuous structural process that converts dietary fat into absorbable units. The transition from large aggregates to dispersed droplets and then to organized transport structures defines whether lipids can be processed and absorbed efficiently. When this sequence is aligned with upstream preparation and downstream demand, fat digestion proceeds with consistency. When it is disrupted, the physical state of lipids limits interaction at every stage, and the efficiency of the system declines. Fat digestion as system stabilizer. Fat digestion influences the stability of the entire digestive sequence because it directly shapes the timing, signaling, and physical environment within the intestine. Lipids are not processed in isolation. Their presence alters how quickly material moves, how enzymes are released, and how the system coordinates the transition between stages. When fat digestion is aligned, these effects create a controlled, predictable flow through the digestive tract. When it is disrupted, variability increases across multiple processes at once. The presence of fat slows gastric emptying through coordinated signaling, extending the time available for upstream processing and ensuring that the small intestine receives material in manageable portions. This controlled release allows enzyme activity and bile delivery to remain synchronized with substrate arrival. The intestine operates within a stable range of input, where breakdown and absorption can proceed without being overwhelmed. Fat therefore acts as a pacing mechanism, regulating the speed of the sequence so that each stage has sufficient time to perform its function. Bile-mediated dispersion of lipids contributes to this stability by organizing the physical state of the luminal contents. When fats are emulsified and incorporated into micelles, they are distributed evenly within the intestinal environment. This uniformity supports consistent enzyme activity and reduces variability in how substrates interact with the intestinal surface. The system processes a more homogeneous mixture, allowing breakdown and absorption to proceed with fewer fluctuations. Fat digestion also influences hormonal and neural signaling that coordinate digestive activity. The detection of lipids in the intestine generates signals that adjust enzyme secretion, bile flow, and motility patterns. When these signals are aligned with the actual state of the substrate, they reinforce coordination across the system. Enzymes are delivered when needed, bile is released in proportion to fat content, and motility is adjusted to maintain appropriate contact time. These responses create a feedback loop that stabilizes the sequence. When fat digestion is impaired, this stabilizing effect is lost. Inadequate emulsification or mistimed bile release leaves lipids in forms that resist enzymatic action. These unprocessed fats alter the physical environment, affecting how other nutrients are handled and how contents move through the intestine. The signals that regulate motility and secretion become less reliable because they are based on substrates that do not match expected conditions. The system shifts from coordinated pacing to reactive adjustment, increasing variability in transit and processing. The influence of fat digestion extends beyond lipid handling alone. Because it regulates timing, distribution, and signaling, it affects how proteins and carbohydrates are processed within the same environment. A stable lipid processing sequence supports consistent conditions for all macronutrients, while disruption in this pathway introduces variability that propagates across the entire system. Fat digestion therefore acts as a stabilizing force within digestion by controlling the rate and organization of the sequence. When emulsification, enzyme activity, and signaling are aligned, lipids contribute to a steady and coordinated flow that supports efficient breakdown and absorption. When this alignment is lost, the system becomes less predictable, and the effects extend to every component of digestion.